Good day! For this video, we are assigned to make an experiment by doing our own pH indicator made from natural sources that can easily find in our home. Also, using our own made pH indicator, we are going to test the pH of some products later on. But before we proceed, let me give you some quick information about the concept and the importance of the pH indicators. pH is the negative log of hydrogen ion concentration in a water-based solution. In 1909, the term pH was first described by the Danish biochemist Søren Peter Lauritz Sørensen, and pH is the abbreviation of the power of hydrogen, whereas P is short for power in German language, and a capital H, which is the element symbol of hydrogen. The importance of pH is it can control the nutrient availability, biological function, microbial activity, and chemical behavior. Because of this, we need to monitor and control the pH level of many things that is needed for a reason. pH measures the corresponding amount of free hydrogen and hydroxyl ions in the water. It measures how acidic or basic the water is. The pH scale is a logarithmic scale that ranges from 1 to 14. If the pH ranges greater than 7, it means the pH is base. If the pH ranges less than 7, it means the pH is acid. And if it ranges from 7 only, then the pH is neutral between acid and base. In measuring pH level, we need an indicator. Now, what is an indicator? Acid-base indicators, also called as pH indicators, are substances that changes color depending on the pH. They are usually weak acids or bases that, when dissolved in water, easily dissociate to form ions. The reason behind the function of an indicator are caused by the dissociation of hydrogen cation from the indicator itself. The importance of indicators is it detects the endpoint of titrations. This means when a certain concentration of the acid or a base is completely neutralized, then at the endpoint, we observe a color change. Now that you have an idea about the concept and importance in this experiment, we will now proceed to see the process during the experimental that was performed by Aina Ragasa, and later on, Lester Guevara will discuss the result observation.
We created a table in which we compared each sample more neatly and clearly. In the experiment we presented, we followed the guidelines to make the outcome more accurate and realistic. So we can see in the experiment that each sample has a base on which we use water. We can see at the bottom of the table the reference we use up so we can compare all the samples. We can measure samples better and more accurately with its help. According to the data we collected, all of the pH ranges, water is the only neutral type of substance. In addition, we also have two types of base, that is the shampoo and the baking soda. Lastly, we have two acids. These are the vinegar and muriatic acid. If we compare their color according to the final product that we have, they will never stray too far from the color that they have, because their base is water, and we could assume that the presence of base or the water was a factor so that there would be no significant change in the color of all samples. Now let's move on to answering the guide questions. For the first question, what color was the indicator for acidic solutions? If we look at our reference again, the color indicator of our acidic solution ranges from bright yellow to dark red. What about for basic and neutral solutions? For the base solutions, the color ranges from olive green to violet. What was the order of your samples from most acidic to least acidic? The arrangement of our samples from acidic to least acidic is as follows: muriatic acid, vinegar, water, baking soda, and shampoo. In the next question, what is the organic compound responsible for the color changes observed? Eggplants materials contain anthocyanins. Anthocyanins are a huge gathering of red-blue plant colors that happen in every single higher plant. These plant shades produce explicit tones and arrangement of various causticity or basicity. They happen for the most part in blossoms and organic products. But on the other hand, they are also available in leaves, stems, and roots. It colors a large number of our food sources. What is the basis for these color changes? The color change of a pH indicator is brought about by the separation of hydrogen ion particle from the actual marker. Review that pH marker are normal colors as well as powerless acids. The separation of the weak acid marker makes the arrangement to change in color. And for the final question, red cabbage is the usual plant source for this kind of experiment. Why do you think experiments still work even though you use eggplants? The molecule in red cabbage responsible for the colors is the anthocyanin. If we go back to what we have said, anthocyanins also exist in eggplants and other plants that are usually our food source. The only important thing in this experiment is that we get color extract from eggplant, which in case we would prefer to get more accurate and realistic results. But the best way to use this experiment is to use the red cabbage, because it will produce more of the color extract that we will need it. To wrap things up, pH is a significant amount that mirrors the synthetic states of an answer. The pH can handle the accessibility of supplements, natural capacities, microbial action, and the conduct of synthetic compounds. Keeping a pH balance in your body is significant to your general well-being. Overabundance of acids in the body can turn into a risky environment that debilitates the body, making the best condition for sickness. At the point when abundant acids should be killed, we utilize the body's soluble stores, leaving us in a debilitated state. Thank you for watching.